My name is Zoe Watson, and this summer I worked in Dr. Zardini-Buzzato's lab to reconstruct lipid topography in brain samples using LC-MS and miniaturized sampling. Lipids are essential molecules involved in cell signaling, structure, and function, and can be affected by many factors, including age, sex, and diet. For this project, I chose to focus on clinical lipidomics, which aims to connect the classes, structures, and functions of lipids to disease processes. Brain lipids play a significant role in neurodegenerative diseases and could provide insight into disease mechanisms. Tissue-specific mapping can reveal alterations that influence neuronal functioning and highlight early drivers of pathology. Now let's zoom in to some specific portions of my project. For this project, we examined the lipid profile of rat brains with prenatal alcohol exposure, PAE. This disease is the leading cause of fetal alcohol spectrum disorder, which results in cognitive, behavioral, and physical impairments. PAE is also known to cause neurodegeneration in adults. The aims of our study were to correlate PAE symptoms with the physical changes in lipids that we see in various brain regions. Lipids were extracted using a modified Folk method. Untargeted lipidomics was performed using LC-MS, and the data was processed using LipidQuest. I first compared all control and PAE samples for 10 different brain regions. Multivariate analysis shows that all brain regions have overlapping lipid profiles, but region-specific analysis reveals distinct disease alterations. I found that one specific lipid, PCO 18025, was changed significantly in 4 out of 10 brain regions, being upregulated in the basolateral amygdala and dorsal hippocampus, and downregulated in the perirhinal and prefrontal cortex. These areas were generally related to emotional-based memory, which have interesting correlations with PAE symptoms. PCs are important in membrane stability and are a good source of second messengers that help transport cell signals. PCO 18025 is involved in distinctly neurological pathways. Plasmalogens have essential structure and function in synaptic signaling and protection against oxidative damage. Inflammation-driven loss promotes plaques and neurodegeneration. We have theorized that this neurodegeneration could be correlated with memory loss in the perirhinal cortex and challenges with executive functioning in the prefrontal cortex. Choline production is a precursor to acetylcholine. Acetylcholine is an important neurotransmitter for the whole brain, but particularly the amygdala. A study showed that after fear-based learning episodes, acetylcholine and noradrenaline were released, increasing the activity of the neurons in the basolateral amygdala. The theory here is that with changes in acetylcholine levels, neuronal misfiring could occur, which would make sense considering that PAE patients have extreme challenges with emotional regulation. Binary comparisons for the effect of PAE on the medial amygdala and nucleus accumbens proved distinctly different from other analyzed brain regions. Unlike all other regions, the total number of lipids shown per lipid subclass increased in the medial amygdala and nucleus accumbens. An overall increase in the number of lipids suggests a change in membrane permeability. This is supplemented by clustered heat maps which associate higher amounts of membrane lipids with PAE than control in both regions. Change in membrane lipids can impact the membrane permeability and the action potentials of the neurons. This makes sense when you consider that PAE is highly comorbid with addiction, anxiety, depression, and behavioral disorders. I detected lipid changes in some unexpected areas. These areas are more commonly associated with aging than PAE symptoms, as they are more strongly related to memory and memory deficits in aging. Significant lipid changes in these areas are associated with inflammatory signaling, synaptic transmission, and immune response, which could be implicated in exaggerated aging degeneration caused by longer-lasting effects of PAE. The results of this research made some significant advancements in the correlation between lipid processes and behavioral processes associated with PAE. We discovered that spatial lipidomics exposes patterns hidden in bulk tissue, linking lipid metabolism to neurocognitive vulnerability. PAE drives region-specific remodeling in the brain, which changes circuits for cognition, memory, and stress, consistent with FASD symptoms. A blood-brain lipid profile comparison also reveals that there is a metabolic effect that PAE has on the body not previously explored. Though we have made significant advancements throughout this project, there is a lot more to be done. I'm grateful to be able to share my research progress with you.